Welcome to this introductory video about StationX's GenePool platform. In the video, I'll explain what GenePool is and run over the basics of navigation and data import, and then I'll demonstrate some analysis workflows that should get you up and running quickly. Once you've watched the video and have logged into your own account on GenePool, you'll find plenty of documentation in our help portal that describes GenePool's features in more detail. GenePool is a cloud-based genome information management system. It's a secure, scalable platform that can handle all formats of genomics data. That data can be imported directly from your computer or via integrations that we've built in from upstream data partners like these. Once you've imported your genomics data, you can attach other information about the patients or samples, say from a limb system. This lets you use all of GenePool's visual filters to easily access your data and lets you use it in your workflows. You can also integrate annotation sources of your own to augment those already provided in GenePool. GenePool's built-in analytics, annotations, and visualizations mean that you can start working with your data in new ways right away. There's also a wealth of curated public content that you can mine or use as a reference to compare with your own data. If you have a standardized workflow you'd like to use repeatedly, you can use GenePool's analysis engine and APIs to create customized clinical reports like the oncology example here, which includes FDA therapeutic guidance and information about available clinical trials. And enterprise features can allow an assigned administrator to regulate access by users and groups of users to different projects, which simplifies collaboration. In summary, GenePool makes it easy for you to store, manage, analyze, and share your genomics data. You can sign up for a GenePool account by going to stationxinc.com and clicking on the Sign Up button. Or if you already have an account, you can go directly to https://mygenepool.com. When you first log in, you'll see the GenePool dashboard. In the center of the dashboard, you can see a newsfeed of recent activity, as well as any new data projects that you may have been given access to. From the dashboard, you also have access to the Help and Support portal, where you can ask a question or search our extensive knowledge base. For example, here I'll look for information on data import. The project area is where you organize your data for further analysis. It's also where you'll find the GenePool reference library, which is a select collection of genomics projects like the TCGA. The account link takes you to various administrative functions, including the ability to connect to third-party partners of StationX where your data may be stored. Some applications require API access to GenePool, and this is where you'll set that up. Also on the dashboard, there's a shortcut to look up gene information. Just type the gene symbol and enter, and the gene card will appear. This has summary description of the gene, some information about its function, structure, and location, and its expression pattern in the body map dataset. Probably one of the first things you'll want to do is import some data. Here's how you do that. Start by naming your project and choosing between gene expression to import count data or sequence variation. Next, you'll be asked about the location of your data. If you have only a few VCF files, for example, you can drag and drop them from your computer. Alternatively, you can directly access and import your data from any of these storage or upstream partner sites via a one-time sign-in. Okay, that's the basics of the GenePool dashboard. Now let's look at a couple of analysis examples to see how you would use GenePool. In this example, I'll demonstrate how to quickly select groups of patients from a large collection of breast cancer RNA-seq samples, and then use an expression comparison analysis to identify differentially expressed genes as potential biomarkers. And when I click on the Projects tab and then look in My Projects, you can see I've already made a virtual copy of the relevant breast cancer data taken from the TCGA reference set that's kept below. Clicking on the project itself, I can see a brief description of it, and then I can look at the samples themselves. You can see there's 1,218 samples in here, each with a great deal of associated clinical information, including family history, overall survival information, some histological data, and some drug and radiation information too. As I mentioned, I'm going to compare two groups of samples. The one group I've already defined to save time, and for the second sample group I'm going to create it based on a different set of criteria. So to create my second group, I'm going to use elements of the user interface to pull up clinical parameters I'm interested in, and use these graphical filters to define my cohort. As you can see, by typing the name of the parameter I'm interested in, I can shorten this list and select the parameter I want. I'm going to select 3 here: the sample type, the estrogen receptor status, and the PAM50 classification. Now, the subset of samples that I'm interested in finding are the primary tumors, estrogen receptor negative, and basal by PAM50 classification. You can see, but just by selecting the wedges on these pie charts, I'm able to very quickly bring down the number of samples from 1218 to the 159 that I'm really interested in. Now all that's left is that I save this group and name it something meaningful: estrogen receptor negative, basal subtype, and primary tumor. And the group that I defined earlier in exactly the same way was also primary tumor and estrogen receptor negative, but it was HER2 enriched instead of basal that I selected. Now, I mentioned I wanted to do an expression comparison between these groups, so I select that, and I can change any of these parameters if I want to, but I'm choosing the defaults for now. So really all I have to do now is choose the 2 groups. One was the HER2 enriched tumors with 51 samples, and the other was basal, 159, and then click Run Analysis. So what GenePool is doing here is retrieving the expression data for all of those datasets in both subgroups, and then performing a differential expression analysis. Now, there's any number of ways I can filter the results on this page, but as I've said, I'm only interested in those things that are most differentially expressed. So on this graph of fold difference, I'm going to remove everything that's clustered around the middle area where there's very little difference in expression. Removing the genes that had very little difference in expression between the subgroups brings us down from 20,000 genes to closer to 400. And one of the next things you could do is ask, are there any classes of genes that are overrepresented in this differential expression? One thing that's useful to do here is to exclude the larger supercategories of genes in the hierarchy to be able to focus a little easier. It's no surprise to see PAM50 near the top of the list, since that's the classification that we use to select our subgroups. But you can see that this list is also enriched for extracellular and cell signaling proteins. Another thing we can do is perform a machine learning technique, unsupervised clustering, on our expression data. The results of this are shown here in an interactive heat map, where the columns represent the samples and the rows are the genes. You can immediately see 2 groupings here, and you'd expect these to conform to the 2 subtypes, HER2 enriched and basal, that we selected in the analysis. You can explore this a bit more by sorting the datasets by the different clinical parameters that we've been using. Using this drop-down menu, you can also pull up the annotations for both PAM50 and HER2 and overlay them. As I mouse over the PAM50 annotations, you can see that there's a perfect separation between the HER2 and basal subtypes. Looking now at the HER2 immunohistochemistry results, there's also good separation here, but not perfect. The co-expression network viewer graphically displays groups of genes whose expression levels are correlated, giving you a sense of how many genes are linked by expression and of how strong these interactions are. There are many other ways you can use GenePool to look for potential biomarkers in an expression comparison, but I'll end this example by pointing out that you can always save your results to retrieve later, and you can also export the results in this table in either Excel or text format if you need to use them in another application. In this example, we'll look at analyzing a trio, comparing the genome of a child with an undiagnosed genetic condition to her unaffected parents, identifying variants potentially implicated in the phenotype. For this example, I've made a virtual copy of a trio of genomes into my project area. They're actually very well studied Ceph genomes, but we can use them to illustrate how we would look for potentially harmful variants. When I look at the samples themselves, I can see there's a little bit of demographic information, but also information on the family structure, which comes from a pedigree or PED-style file. Moving on to the analysis, I will select the trio analysis, and you can see it's already pre-configured with the information from the PED file, and there are also some default parameters that are selected. I won't change those. The only other thing I need to do now is select the samples, which in this case is all 3 samples in the project, and then run the analysis. At this point, GenePool very efficiently retrieves the variants in the genomes for these 3 individuals, and then associates them with genes that they occur in, and information about the effect of that variant on a protein coding region, if it is in a protein coding region, that is, and the genetic pattern in this family, whether it's dominant, recessive, or de novo. GenePool also attaches annotation information from whichever databases are available and configured for this analysis. You can easily modify these. For example, let's say that I know the exact subpopulation of this group. I can select their allele frequencies rather than the more general population. So in this case, I'm going to select non-Finnish Europeans. We'll keep the ClinVar and the Clinical Genomics database annotations. As you can see, there's many other annotations you could select from, for example, the FDA biomarkers or the American College of Medical Geneticists recommendations. To identify potential variants as candidates for causing a genetic condition, the first thing I might do is restrict my list to those variants that modify a protein sequence. If I select here those that are predicted to cause a high or a moderate impact, you can see that that reduces me down from 6,300 variants to around 2,300. The second thing I might do, since the assumption is that this is a rare genetic condition, is to use my exact frequencies for non-Finnish Europeans and restrict them to around 2% or less allele frequency. In other words, the remaining variants in the list are either not seen in this subgroup of the population or are not expected to be seen at any more than 2% allele frequency. Very rare. The last step I'll do in this analysis is to look at the ClinVar database and select any variants that have been identified as being pathogenic. This effectively reduces my initial list of variants down to just 4. As we review the genetic data here, we can see that all of these variants are inherited in a dominant fashion and that the child is heterozygous. But only 2 of these diseases are expected to be inherited in a dominant fashion based on the Clinical Genomics database. Now let's say this child had neuromuscular symptoms. Maybe this is the variant on this list that's most likely to fit the clinical situation. I can then drill down further and explore the underlying sequence data supporting this reported variant, just to make sure that indeed it's heterozygous, which it looks to be the case. And I can further explore what the effect on the protein sequence is, in this case, changing a threonine to methionine. And there's a lot more information here to explore. Once I'm happy with my list of variants, I can save my results as a report, name it something meaningful, and attach it to my project so that I can come back to it and retrieve it at any time. Well, alright. Hopefully I've been able to demonstrate to you how easy it is to use GenePool's trio analysis to identify potentially harmful variants. Thanks very much.